Hi everyone, I'm Nithika Vivek and welcome to Module 2E, How to Design a Winning Research Project. I participated in ISEF and I'll be one of your instructors for this course. For my ISEF project, I developed a multimodal deep learning model that visually identifies melanoma lesions by integrating structured metadata. That experience taught me the importance of having a strong foundation in research design, which is what we'll be going over in this slideshow. We'll also dive into essential strategies for conducting a successful research project. I'm excited to share these tips with you, and I hope you find them valuable. So first, we're going to start off with key variables to identify. The first variable is the independent variable. This is the variable you're trying to manipulate to measure an effect. So let's just think of a sample scenario. For example, let's say you're trying to answer the question, how does the amount of light affect the growth of a plant? The independent variable in this case would be what you're changing to measure the effect it has on the growth of the plant. For example, one group of plants will receive 12 hours of light a day, and another group will receive 6 hours of light per day. So this is the variable you're manipulating to measure how it affects the growth of the plant. And this leads us to the second one, which is the dependent variable, which is the variable you are measuring the change of. So in this case, this would be the growth of the plant, because you're trying to measure how does light affect the growth of the plant. So it depends on how much light the plant receives and will be measured regularly throughout the experiment. And then the last variable that's important is the control variable. So these are variables where the factors will be kept the same for all plants to ensure that the reason that the growth of the plants is different is because of the light and only the light. Examples of control variables would be the type of plant, the amount of water each plant receives, the type of soil, the size of the pots, the temperature of the environment, or the location of the plant. A key thing to note is that in science, it's not just about discovering new things. It's about communicating those discoveries effectively. Staying organized throughout your experiment is crucial because it ensures that your results are reliable, accurate, and reproducible. And producing reliable and reproducible results allows other scientists to replicate and therefore verify your experiment, which is a key thing that people do when you're trying to get your research published. Before we get into an example of a research experiment, I first want to talk about ethical considerations. So the first important one is informed consent. This means that the participants fully understand what the study is about, any risks involved, and what they can do if they want to withdraw anytime. The next one we have is confidentiality and privacy. Researchers must protect the participants' personal information, which keeps the data secure and only accessible to you, the researcher, and your team. The privacy ensures that participants are safe and respected in this environment where they might feel vulnerable because they're technically being experimented on. The third one is minimizing harm. This one is really crucial because we must take steps to reduce any potential physical or psychological harm to patients. This means careful planning and readiness to provide support if any participant feels unsafe or in danger during an experiment. The fourth one is avoiding deception, which is about being honest. If deception is necessary for the study, like you're hiding some information from the participant if you're performing a psychological experiment, it must be justified and participants should know that this has happened to them afterwards. And the fifth one is animal safety. This is mainly for studies involving animals where we must ensure humane treatment using alternatives to animal testing whenever possible. And the sixth one is transparency and honesty, which means being open about the research process, funding sources, and reporting results truthfully. It's really important not to fabricate data because most people are caught in the future, and as a researcher, we're all trying to contribute something to the research environment, and fabricating data is not the way to do that. So now we'll go through an example setup. So first, we have to define our objective. So the objective would be to develop a new building material that is sustainable and environmentally friendly by using recycled waste. And then now we're going to define the questions. which makes it more structured on what we have to follow and what we have to answer throughout the experiment. So 2 questions that could be defined in this particular experiment could be, what is the environmental impact of the new material? And is the new material cost effective compared to traditional building materials? So notice that these 2 questions directly contribute to answering our overall objective. Afterwards, we form a hypothesis. For example, this would be like, building new material will have superior mechanical properties compared to conventional methods, making it both strong and sustainable. So this hypothesis, if you can see, answers the research questions and objective, but it's based on background research. So hypothesis is not just a guess. It's on background research that you as a researcher do to decide on your objective and predict what will happen with your variables, which takes us into the different types of variables we will be manipulating or measuring in this experiment. So the independent variable would be the type of recycled waste used, because this is what's manipulated to measure the impact on the material's properties. The dependent variable would be the mechanical, thermal, and environmental impact of the material. These are what we'll measure to assess performance. And then the control variables would be variables that may change the impact this has on the dependent variables, such as temperature, humidity, dimensions of the material samples. And by keeping this constant, we can ensure that any changes in the dependent variable are due to the type of recycled waste and not other factors. So continuing with this example, now we'll define methods. Defining methods before you do the experiment is especially important because it helps ensure that you know as a researcher what you're going to do in each part of the experiment. So we will first begin by selecting different types of recycled waste. These materials could then be mixed or poured into molds to create test samples. Once these samples are prepared, we can test them under controlled conditions to ensure consistency. Afterwards, we'll go to testing, which is where we'll test the samples for compressive, tensile, flexural strength to determine their mechanical properties. Notice how mechanical properties was one dependent variable, so the testing we're doing in this part directly contributes to a dependent variable. The next dependent variable we have is thermal conductivity, which we could assess by seeing how well the material insulates against heat. Afterwards, we also have to tackle the environmental impact, where we'll calculate the total carbon footprint of the material. An additional variable you might want to measure in this experiment is the cost analysis to see if this is environmentally and economically feasible. Then we go on to data analysis. So in data analysis, you can analyze the data through statistical tests such as ANOVA t-tests to identify significant differences between samples. Regression analysis will help us then develop equations and models based on material composition. So in other words, the whole point of regression analysis is to define an independent variable as a function of the dependent variable, which is basically producing an equation to connect the 2 variables. We will then compare different metrics, including strength, thermal properties, and cost, to determine which material performs the best. So there are many different ways you can collect data. The previous experiment was one way we collected data, and this was a wet lab study because you are actively performing something on the dependent variable to measure what effect the independent variable has. However, other types of collecting data include surveys, which is good because it reaches a lot of people and is cost effective. Interviews, which allow for deeper understanding, provide qualitative data instead of quantitative because you're having a conversation with the research participant. Observation provides real-time data and is not intrusive. Case studies give detail and context and hypotheses. Wet lab establishes a cause and effect relationship. Ethnography gives insight into social phenomena, and secondary data is data you get from online, pre-processed or manipulated in some way, and then find something useful out of that data. Experiments can be one of these or a combination of these, and it depends on what variable you're trying to measure, which is what techniques you'll take to collect the data. There are many different data analysis tools that you can use throughout the experiment. So One tool is RStudio, which is used for data visualization. An example of this is when researchers want to visualize climate change data over time in graphs. RStudio will make it easy to detect these changes through a plot called ggplots. ggplots plot the data in an organized fashion and a user-friendly way to establish a relationship between two variables. The next data analysis tool you could use is pandas, which is used to pre-process machine learning data to improve model accuracy, such as normalizing and augmenting data to reduce overfitting. I used pandas in my ISEF experiment because I was dealing with machine learning data, and this data is pre-processed differently than how data would be in, like, let's say a wet lab experiment. Another data analysis tool is Excel spreadsheets, which is used to store large amounts of data in tables after wet lab data collection. And then the fourth way is PyMol, which is used to visualize protein structures to see which structure will result in the best binding with a certain molecule. So that's the end of this slideshow, and I hope you got some useful information. Thank you.